So before the panel, our last speaker is Professor David Olson from UC Davis. Welcome. David, can you hear us? Can you hear us?
Yes, I can hear.
Yeah,
yes, I can hear you. Just give me 1 second to share my screen.
Sure. The talk is titled Mechanisms of Psychedelic-Induced Neuroplasticity. Thanks, David.
Does this look fine to everybody?
Yep.
Good, great. All right, let's get started. So thank you for the invitation. I'm sorry I couldn't be there in person. So my name is David Olson. I'm the director of the Institute for Psychedelics and Neurotherapeutics at UC Davis. Before we get going, I also have to tell you that in addition to my academic appointments, I'm also a co-founder and the chief innovation officer of Delix Therapeutics. But everything I'm going to tell you about today was work that was done in my academic laboratory. So our group is really interested in finding new treatments for so-called stress-related neuropsychiatric diseases. These are conditions like depression, post-traumatic stress disorder, and substance use disorder. And a hallmark of all of these illnesses is the atrophy of key brain regions, in particular, the prefrontal cortex. And the prefrontal cortex is really critical because it controls motivation, fear, and reward. And a hypothesis in the field has been that if you could identify chemical stimuli that can induce the growth of cortical neurons, such as the growth of new dendrites, or if you zoom in on one of these dendrites, you'll see the growth of these dendritic spines. If you can promote that type of growth, you might be able to achieve a few different really interesting results. Number one, you might have a rapid acting response. Number two, the response might be sustained over a long period of time. And number three, the treatment might actually work across indications because the PFC is such a critical hub. And so when we're looking for these types of chemical stimuli, one of the first molecules, probably the best known molecule that has this ability is this molecule here on the left, this dissociative anesthetic called ketamine. And so here I'm showing you some cultured cortical neurons that were treated with ketamine. And you can see that they have increased number of these dendritic branches, and these little green dots are representing synapses. So they also have an increased number of synapses. So ketamine promotes both neuritogenesis and synaptogenesis. And now I'm showing you some in vivo work that was performed by the late Ron Duman and the late George Akajanian. What they were able to show is that chronic stress can lead to that cortical atrophy that I told you about, the loss of those dendritic spines in the prefrontal cortex, and that really manifests as synaptic deficits that you can measure with traditional techniques like electrophysiology. But what is really remarkable is that a single dose of ketamine within 24 hours can rapidly regrow those lost dendritic spines in the cortex and reestablish synaptic connectivity. Now, I think ketamine is super exciting for the field because it's pushing us in a completely new direction, a direction that is more in line with a healing-based approach, trying to repair these damaged neural circuits. But the issue with ketamine, there are many. One of the issues is really its kind of lack of circuit level selectivity. The primary target of ketamine is the NMDA receptor, which is found broadly across the entire brain. And so when we got into this area, we were thinking very deeply about what receptors might be selectively expressed in the specific neurons and brain regions that we really care about for treating these stress-related neuropsychiatric diseases. And that really led us to study the serotonin or 5-HT2A receptor. Here I'm showing you the expression patterns of the 5-HT2A receptor across the rodent brain. And you can see it's heavily expressed in these layer 5 pyramidal neurons of the cortex, with more expression in the anterior parts of the brain, less expression as you move towards the back of the brain, and much lower expression in structures like the hippocampus or the striatum. And so one of the first experiments that we did was simply to take cultured cortical neurons, treat them with psychedelics, and look at neuronal morphology. And you can see that compared to the vehicle-treated neurons in the middle, the neurons treated with the psychedelics, DMT and LSD, have substantially more complex dendritic arbors. And if you do some super resolution microscopy and zoom in on one of these dendrites, you'll also see an increase in the number of dendritic spines and these filopodia-like structures. Now, there's a number of ways to quantify changes in cortical neuron structural plasticity. I'm going to briefly tell you about one way that we use fairly regularly. It's a very simple method. It's called shoal analysis. The idea is you just draw these concentric circles of increasing radii starting at the soma of the neuron, and then you simply count the number of times the dendritic branch crosses one of those circles. And you can produce these really, really beautiful shoal plots like the one here on the right, where on the x-axis are the radii of those concentric circles, and on the y-axis is the number of crossings. But typically the way we like to represent the data is simply looking at the maximum number of crossings for a particular drug. And so for the vehicle control, it would be about 7 here. For the DMT, it would be about 11. So looking at these Nmax plots, what we can see is that psychedelics from 3 distinct structural classes, ergolines like LSD, tryptamines like DMT, and amphetamines like DOI, they're all very good at promoting cortical neuron growth. Now, all of these early experiments that we were doing, we were treating cultured cortical neurons for 72 hours. And we know that in vivo, that's really not what happens. In vivo, you give a psychedelic drug, it rapidly gets into the brain, and then it's rapidly cleared. But there's very long-lasting effects. And so we were very interested to see what is the shortest stimulation period that we could use that would produce long-lasting changes in neuronal structure. So instead of stimulating for 3 days, we started pulsing with for 24 hours or an hour or maybe just 15 minutes. And then we take the drug away and then give the neurons time to grow and look for changes in morphology. And what we find is that even with these really short stimulation periods, just 15 minutes or an hour, you can get very profound increases in cortical neuron growth. But the trick is you have to give the neurons time to grow. If you just stimulate for an hour, fix and stain, you see absolutely no change in morphology. But if you stimulate for an hour, give the neurons a couple of days to grow, then you can see pretty large differences. So to demonstrate this effect in vivo, we relied on using a psychedelic that is rapidly cleared from the body, dimethyltryptamine. The half-life of DMT in a rodent is only about 10 minutes. And so for this experiment, we gave a single IP injection of DMT to rats. We then waited 24 hours, a time period when all of the drug has been cleared from the body. And we then sacrificed the animals, look at neuronal structure via Golgi staining, and function via electrophysiology. And what we find is really interesting. We find that even after the drug has been cleared from the body, we get this long-lasting increase in dendritic spine density, and that's accompanied by functional effects, including increases in both the amplitude and the frequency of these spontaneous excitatory postsynaptic currents. Now, since we published this work way back in 2018, it has been replicated and extended by a number of groups across the world. But I want to focus on one study in particular because I think it's really, really interesting. This was work done by Alex Kwan's group back when he was at Yale. He has since moved to Cornell. But Alex was really interested in seeing just how long do these effects last for. So he gave a single dose of psilocybin to male and female mice, and he finds that psilocybin induces increased density of cortical of spines in the prefrontal cortex that lasts for over a month, which is just really remarkable to me. So at this point, we're very interested in trying to understand the mechanism by which psychedelics promote this cortical neuron growth phenotype. And so we started with just some selective pharmacology. So the very first thing we did is we tried to block the serotonin 2A receptor because it's well known that the serotonin 2A receptor mediates the hallucinogenic effects of psychedelics. And if you give a 5-HT2A antagonist to people, you completely block the subjective effects of those drugs. And what we find is that the 2A antagonist can completely block the effects of psychedelics from the 3 different structural classes. And then the next thing we did is we tried to look at some downstream kinases. These are 2 kinases that were implicated in the plasticity promoting effects of ketamine. The first is called TrkB, which is the high affinity receptor for brain-derived neurotrophic factor. And again, we find that if we block TrkB, psychedelics are no longer able to promote cortical neuron growth. And the same is true of this downstream kinase, mTOR. mTOR is really important for producing all of the proteins necessary for plasticity, all of the structural proteins that allow neurons to change their shape, and all of the ion channels that allow neurons to change their synaptic weights. So at this point, this was decent evidence, but we realized that the antagonist that we were using to block the serotonin 2A receptor is not perfectly selective. It hits a couple of other receptors. So to be absolutely certain that the 5-HT2A receptor was the target, we relied on genetic models. And so here we used animals that have had the 5-HT2A receptor genetically deleted. And we can see that in wild type animals, the psychedelic 5-methoxy-DMT produces a very robust effect on cortical spine growth, but the effect is completely gone in the 5-HT2A knockout animals. We're not the only group to observe this. Javier Gonzalez-Maiso's lab has seen a very similar result with a different psychedelic, an amphetamine type psychedelic called DOI. Now, we also looked at neuronal function. And so here again, you can see that in the wild type animals, you get this increase in the frequency and amplitude of these spontaneous excitatory postsynaptic currents. But in the 5-HT2A knockout animals, the effect is completely gone. So now we wanted to see if the 5-HT2A receptor might be involved in the behavioral effects of psychedelics. And so for this, we relied on a couple of different assays. I'm going to show you first this one, which is a modified version of the forced swim test. And we've modified this in such a way that traditional antidepressants like SSRIs are not very effective, but these next generation antidepressants like ketamine tend to be effective. And so the way we do this is we run a forced swim test on the first day to just do two things: establish that all groups have exactly the same level of immobility, and also provide some additional stress to the animal. And then we give our dose of compound. We do a behavioral test, immediately after, just to ensure that we've engaged the target appropriately. And then 24 hours later, after the drugs have been cleared from the body, we then do our assessment of efficacy. And what you can see is that when you give 5-methoxy-DMT on its own, you get this decrease in immobility, this antidepressant-like behavioral response. But if you use the 5-HT2 antagonist ketanserin, you can completely block this effect. Now, a forced swim test is probably a little more relevant to motivated behavior. So we wanted to look at an assay that is more related to anhedonia. And for that, we relied on the sucrose preference test. So mice, much like myself, will prefer a sugary drink over just plain old water. And you can use this preference as a measure of anhedonia. And so the way we do this is on the first day, we give them a choice between sucrose and water. And you can see that they all prefer the bottle of sucrose. We then can induce an anhedonia-like phenotype by giving some kind of chronic stress. And in this case, we're giving chronic administration of corticosterone. And then following 10 days of core administration, we do another sucrose preference test. And you can see that in both the wild type animals and in the 5-HT2A knockout animals, there is this drastic reduction in their preference for the sucrose bottle. On day 13, we give a single injection of psilocybin. And then 24 hours later, we measure sucrose preference again. And you can see that in the wild type animal, psilocybin causes a very nice rescue of this sucrose preference. But you don't see that same effect in the 5-HT2A knockout animals. So at this point in time, we're feeling pretty confident that the 5-HT2A receptor mediates the effects of psychedelics on structural cortical neuroplasticity, and also maybe some of the antidepressant behavioral responses. But there was a piece of data that had us just scratching our heads for the longest time, and it's this one. And it was the fact that this molecule right here, serotonin, could not promote cortical neuron growth. And that was really surprising to us because I just spent the last 10 minutes telling you that the serotonin 2A receptor was responsible for psychedelic-induced structural neuroplasticity. And so it didn't really make a ton of sense until we got this particular structure-activity relationship study finished. We started making some small modifications to serotonin-like molecules over here on the left. And we noticed that as you started to add methyl groups, going from one methyl group to two methyl groups, you saw a much bigger effect on cortical neuron growth. And you can see that here in these images, but it's very easy to see this with these show plots. So going from this molecule here, tryptamine, to N-methyltryptamine to dimethyltryptamine, you see very big increases in cortical neuron structural plasticity. So then what we did was we tried to correlate the activity of the 5-HT2A receptor when it was heterologously expressed in a non-neuronal cell like a HEC cell with cortical neuron growth. And we saw absolutely no correlation at all. If anything, we saw a slight negative correlation. But when we tried to correlate the lipophilicity of the drugs, how greasy are the drugs with their ability to promote cortical neuron growth, we saw a very nice positive correlation where these very polar drugs like serotonin and tryptamine could not promote cortical neuron growth, but these non-polar drugs like these psychedelics are very good at promoting cortical neuron growth. And that led us to the hypothesis that maybe the reason that serotonin cannot do the same thing as something like DMT is because maybe it can't actually access the target. The 5-HT2A receptor is a G protein coupled receptor, and canonically, many GPCRs exist on the plasma membrane, and they're meant to interact with molecules at the plasma membrane and translate that into intracellular signaling cascades. But in this case, we thought maybe the 5-HT2A receptor was on the inside of the cell. And if that was the case, serotonin via a zwitterionic compound is too polar to passively cross the nonpolar membrane and access the target. So the first thing that we did is we simply wanted to look for, where is it located in a neuron? But before we did that, we wanted to see where is it located in just a hex cell. And so here I'm showing you the expression patterns of the 5-HT2A receptor, a different GPCR, the beta-2 adrenergic receptor, and then this is just the CFP control. You can see that the CFP goes everywhere in the cell, but the two GPCRs are localized where you would expect them, kind of on the plasma membrane with some intracellular as well. This white line is just a plasma membrane marker. But when you look in neurons, in cortical neurons in particular, you see something very different. You see that for the 5-HT2A receptor, most everything is localized intracellularly, but the beta-2 adrenergic receptor still has lots of receptors on the cell surface. And so these are live cell imaging. We also wanted to do this doing fixed cell imaging so that we could try to localize exactly which subcellular compartment the 5-HT2A receptor was found in. And I should note that, this was mainly a confirmation study here. We're not the first to show that the 5-HT2A receptor is intracellular in pyramidal neurons in the cortex. This has been shown by many, many other groups over the course of, maybe 20 years or something, but no one really understood what was the functional consequence of this intracellular localization. So when we tried to do our subcellular localization analysis, we found something really interesting. And that is that the 5-HT2A receptor co-localized extremely well with markers of the Golgi. If you look in a hex cell, you don't see the same thing. So here is 5-HT2A receptor expression. You can see there's a bunch of 2A that is not localized with the Golgi marker. So it's very different in cortical neurons versus hex cells. So next, to try to understand this a little better, we developed some new molecular tools based on these membrane permeable compounds here in green. So we've got the membrane permeable agonist DMT and psilocin, and the membrane permeable antagonist ketanserin. And we just converted them to their membrane impermeable versions in purple by methylating DMT or putting a phosphate group on psilocybin or methylating ketanserin. So all the compounds in purple are permanently charged, and so they cannot passively cross a nonpolar lipid bilayer. So then what we did is we ran the assay under two different conditions. One is just normal, plain old cortical neurons. The other one is with a little bit of electroporation. And with electroporation, you can apply a voltage to transiently open pores in the neurons. And what that does is it allows the membrane impermeable compounds to cross the membrane, the pores close, and then the impermeable compounds get stuck on the inside of the cell. And when we do that, we see something really interesting. Without electroporation, we get the effects that you would expect. DMT and psilocin, the two membrane permeable agonists, are very good at promoting cortical neuron growth, whereas the impermeable agonists, TMT and psilocybin, cannot. Similarly, the membrane permeable antagonist, ketanserin, can block the effects of DMT, but the membrane impermeable antagonist, methylated ketanserin, cannot block the effects of DMT. But when you electroporate the cells, you get the opposite. So now we can see that TMT and psilocybin can promote the growth of cortical neurons, and methylated ketanserin can block the effects of DMT. So these are just some neuritogenesis studies. We also did this with spinogenesis, and we saw the exact same effect. The membrane permeable compounds promoting growth, the membrane impermeable compounds not promoting growth. So at this point, we reason that if it really is all about the location of the receptor, serotonin should be able to do the same thing that a psychedelic can do if we can just get it to the target. And so normally the serotonin transporter is expressed on presynaptic serotonergic neurons. It's not expressed in postsynaptic pyramidal neurons of the cortex. And so for this experiment, what we did is we sparsely transfected the transporter so that we could import serotonin into a select number of neurons. And I love this experiment because within the same culture, you have some neurons that can import serotonin and some neurons that cannot. So looking here at the vehicle side, we've got two neurons that are sitting side by side. The one on the left expresses the transporter, the one on the right does not. Similarly over here, again, two neurons side by side, the one on the right expresses the transporter, the one on the left does not. And so then we can treat the cultures with the various compounds, and we find that in both genotypes, the vehicle doesn't really do anything. And DMT promotes the growth of both types of neurons, both genotypes, presumably because DMT can just passively cross the membrane, so it doesn't require the transporter. But what's really interesting is that serotonin can promote the growth of the neurons that can import serotonin, but it cannot promote the growth of the neurons that cannot. And so this next study was, a partial replication of the last one. So you can see here the same effect, serotonin promoting the growth in SERT-positive neurons. But then we wanted to assess the effects of the 5-HT2A receptor and the transporter. So the other thing that we did is we tried to block the effects of the 5-HT2 antagonist ketanserin. And you can see that the effects are completely blocked. But then we also use citalopram to block the transporter itself. And again, it seems that serotonin requires the transporter. So this study really indicates that serotonin requires the transporter to get inside the cell, and then it requires the 5-HT2A receptor to produce the growth effects. Now, if you just change serotonin for a membrane permeable compound like DMT, you see something very different. Again, DMT can promote the growth of both genotypes. Ketanserin can block the effects of DMT in both genotypes, but citalopram cannot block the effects, again, because DMT does not require import through the transporter. So these were all neuritogenesis studies. We ran the exact same studies in spinogenesis, and here are very similar results. Again, no effects of the vehicle, DMT promoting spinogenesis in both genotypes, but serotonin only promoting spinogenesis in SERT-positive neurons. So to test this hypothesis in vivo, we relied on a genetic strategy. So we used a virus to express the serotonin transporter on the postsynaptic side on those cortical neurons. And then after 3 weeks to allow the virus to express, we then gave a single injection of parachloroamphetamine, a selective serotonin releasing agent. And then 24 hours later, we come back and we look at dendritic spine density. And what we find is that only in the neurons capable of importing the monoamine do we see this increase in cortical spine density. Now, if increases in cortical spine density are important for the antidepressant-like behavioral effects, we would expect to see something very similar using this paradigm. And so what we did is, again, express the serotonin transporter using a virus. We then ran a novelty-induced locomotion assay just to make sure there was no differences between the genotypes, and there wasn't. We then do our forced swim test, the pretest, again, showing there's no difference between the two genotypes at baseline. We then gave a single dose of PCA, came back 24 hours later, and what we find is that we get this antidepressant-like effect only in the animals that have the serotonin transporter on the postsynaptic side and can import the released serotonin. So in summary, what I tried to tell you about today is the fact that psychedelics can promote cortical neuron structural neuroplasticity through the activation of a particular intracellular population of 5-HT2A receptors, really highlighting the role of location bias in their signaling. And then finally, I'd like to thank these groups for funding. And most importantly, I'd like to thank all the incredible students that I get to work with every day. I've highlighted the students or I've highlighted the names of some past students whose work I highlighted today. But you can see all the people in white. These are current lab members who, unfortunately, I didn't have a chance to talk about a lot of their work, but maybe sometime in the future. And with that, I'd be happy to answer any questions.
Maybe.
Hello, thank you for your talk. I don't know, I've been heard?
I can hear you.
Okay, I couldn't see you, so I didn't know. Okay. Yes, well, your paper is incredible. It's been a hot topic this last year for many discussions. And well, before reading your work, I have read the work of Dr. González Maeso and also Dr. David Nichols, which in the early 2000s already had this question of why some 5-HT2A agonists do elicit these psychedelic responses, and while others do not, like serotonin itself. And their work was more, well, they discovered like that bias agonism of the 5-HT2A receptors in the cytoplasmic membrane was very important. And actually there was a different engaging of signaling molecules depending on if on the nature of the agonist of this of 5-HT2A receptors in extracellular receptors. Yeah, you've got, even they showed that some signaling cascades that are related to plasticity were engaged, like the MAP kinase cascade or the EGR transcription factors. So, I mean, I was wondering, how would you integrate all this previous research on the field with your team's discoveries? Like, is it all about these intracellular receptors, or maybe there are like different mechanisms that are acting all together, and these are like different parts of the same story? So,
yeah. So first thing I want to say is that we were just talking about one very specific population of 5-HT2A receptors, just the ones that are in the cortical neurons of the PFC. There's 5-HT2A receptors in lots of other brain regions, and their signaling mechanisms could be completely different. The other thing is that the 5-HT2A receptor is well known to exhibit functional selectivity. What the consequence of that functional selectivity is still very much an open question. A lot of those functional selectivity studies were done in things like HEX cells, where the signaling might be different than in various neuronal populations. And that might be different depending on the brain region. And the other question, so I guess the question is G protein versus beta arrestin. When people talk about functional selectivity, that's typically what they're talking about, although there's more aspects to functional selectivity. I will say that if you just look at the biased signaling of psychedelics in a HEX cell, and then you try to figure out which ones are hallucinogenic or not, there's really not much of a correlation. There are G protein biased ligands that are hallucinogenic. There are beta arrestin biased ligands that are hallucinogenic. And I think the field is now coming to the point where it's generally accepted that G protein and beta arrestin differences, it might not play much of a role. And it seems that G protein signaling is probably the bigger, more important effect. But I want to, you pointed out the fact that Dave Nichols said this, but Dave Nichols also, which is really intriguing, has some old work, and it was really what inspired a lot of this, that he demonstrated that the lipophilicity of psychedelic drugs tracked really, really nicely with their hallucinogenic potency in people. And I think he was just hypothesizing that had to do with their ability to get into the brain, but it might also be their ability to just cross membranes, period, and access this intracellular target. And so molecules like LSD are very interesting because their pKa's are very tightly controlled, such that a large proportion of the molecule exists as both the protonated and the deprotonated state at physiological pH. And that allows them to very easily cross membranes.
Thanks, David. Did you want to add a comment to this question? You had your hands up, or do you have a question?
So can you hear me now? So
Yes.
I have a question and a comment. So the question is actually related to the previous one, but specifically, so now you have these pyramidal cells that either express serotonin transporter or not. So what is the difference in the signaling of those receptors that are at the surface or inside? I mean, why would they be different? And that's the question. And the comment, okay, the comment is that there's been work by Patricia Gaspar a long time ago, but largely ignored. She showed that during the early development, many pyramidal neurons actually express serotonin transporter during the time of growth. So I'm just wondering whether, whether this could be related to your work, so that serotonin might be actually acting as a sort of more effectively.
Yeah, no, it's a really good point. And so, in early development, there's a ton of plasticity that's going on. And if there is transporter expressed on the cortical neurons at that time, then serotonin could be a very effective ligand for that system and might be responsible for the early structural plasticity that you see. But in the cortical cultures that we do, we don't see any expression of the transporter unless we overexpress it. At least it's not expressed at high enough levels that it could really import a lot of serotonin. And then the first question was about the signaling. The answer to that is it's unknown. We don't really know if these different populations of receptors signal differently, or if it's just about which ligands are able to access them.
Yeah, but I mean, if they produce different effects, they should actually have a different signaling, you would expect. Wouldn't you?
Well, it could also be just the quantity. It could be that, there's a small number on the cell surface, so you don't elicit a full response. But there's a lot more of them intracellular. It's possible.
Thanks very much. I think that concludes all the speakers. Thank you very much. I would like to invite everyone. So for the panel discussion, I would like to invite our speakers up here. And David, you can still hear us, right? Can you see us as well?
Yes.
Yeah, perfect. Okay. So Kim, Rob, Dietmar, and Torben, please come up front. And there are many chairs in front, so now whoever has been sitting on the floor can grab chairs. So we are waiting for Dietmar? Yeah. We can start a little bit with a general question. So, am I missing Dietmar? Yeah. Okay. No, probably is. So, the general question, I think this is for all the speakers in the panel. What do you think is the grand challenge or the main hurdles in combining or bridging the gap between basic science and clinical research in psychedelic neuroscience? It's for all of you in the panel. So now Dietmar is here. So the question is, what is the grand challenges or the difficult hurdles in bridging the gap between basic research and clinical research in psychedelic neuroscience?
Well,
this is like to all speakers.
I always feel like I'm going to state the obvious. one challenge is those of us who are working in especially rodent models is we need to develop more sophisticated behavioral assays that hopefully, and it's very challenging, are going to be more predictive of the behavioral effects and perhaps even the subjective effects in human beings. I mean, I'm looking, David, how's your baby? Good. You know, David presented, you know, nice work on the force swim test, but everybody in the field knows that's a primitive assay for trying to assess motivation. So there are better ways. I mean, I'm just using that as an example of trying to assess motivation and decreases in motivation in stressed animals and improvements in motivation. There are better ways of testing hedonia or anhedonia. I mean, the challenge is they're more labor intensive. So, I mean, and again, I feel like I'm stating the obvious. I think David's work, among others, is really valuable because, you know, everybody in this audience knows the huge question in the topic of the therapeutic potential of psychedelics is, do you need the subjective experience or not? And David is appropriately pushing the idea with his brilliant chemistry that perhaps the subjective experience is not necessary, that you can create drugs that don't cause a head twitch assay in mice, so I think the types of tools David and others are creating are going to be incredibly valuable, you know, and it's one of the reasons I started my talk. I like drugs as probes of brain function because you can give them to our rats and mice and you can give them to human beings and you can go back and forth. What else can we do to build better bridges? I mean, obviously, circuit analyses, you know, doing the best we can, trying to figure out in a more sophisticated way what functional connectivity changes that are, you know, using fMRI measurements in human beings. What does that actually mean? And how do we mimic that and model that in animals? And I can go, sorry, I'm going to shut up in a second. I've thought a lot about this, and there's a lot of, I think, bridges that can be built that are not specific for psychedelic science. They're going to be useful for broad areas of psychiatric neuroscience, but I'm going to let somebody else talk.
Dietmar wanted to comment?
Could I just follow up on what Rob said real quickly before we move on? I just want to say I agree completely with what Rob said, but I also want to emphasize that rodents are not people. They will never be people. And so to develop, you know, the behavioral tests are going to be challenging. It's always going to be challenging to get really good behavioral tests that mimic a human condition. When I was a postdoc, I was trying to develop a genetic mouse model of schizophrenia, which is probably the dumbest thing ever because they don't have the brain region that is mainly impacted in human. It's a uniquely human condition. So what I think could be really impactful for the field is the development of translatable biomarkers. If we understand the circuitry better in rodents, then maybe we can just go directly and look at circuitry in humans, whether that's some kind of imaging method, EEG, who knows? But I think that will help the translation.
Yeah, can I, before you, just to add something, or something I also thought about. I've done a lot of MDMA research, and one thing I always saw in the literature is the translatability of the dose. And that is my big question. How do you determine the dose? Because when you showed your data on MDMA, you said 7.5 milligrams per kilogram. I was already calculating. Wow, massive dose. So why is that then a low dose, and why do these animals not feel it? I also know that there is a difference in metabolism, and it also is a difference whether you use a mice or a rat. So, yeah. So how do you determine it?
I think Gita Knudsen would say that you should do it with receptor occupancy using PET ligands, right?
So I would say there's not a single cure, single answer to this question. So first, let me say I'm a big promoter of basic research. I think also without any thought on translation. I think this is important that people also have the freedom to just do basic research, and often then things pop up. And I think there it becomes important. So one thing I would like to bring up here, I was asked to coordinate a big grant, a collaborative research grant. This is how funding in Germany often is organized. And this was on translational neurosciences. And now 15 years later, there are a few things where we have improved. So really talking to each other sounds so simple. I mean, there's not one way to do it, but we put in, my very simple idea in the beginning was trigger some interest for strong natural science people for medicine. And the grant I'm coordinating, they are really very strong biophysicists, physicists, biologists, guys who have discovered optogenetics and so on. So to interest them for medical issues. One good example was we hired somebody from genetics. His main model was Drosophila in the beginning. Now, 15 years later, he has a clinical study on autophagy and aging, which was kind of impressive. And then what we had, a couple of things. What is the main issue actually that we are not able to translate? Is it the model? There are many different animal models, but for example, the robustness sometimes of data. So this is one guy, a clinical guy I work with a lot from neurology. He said, well, let's have a reproduction really of the study. And it turned out, I think NIH, there were 20 studies, looked good, and then they were reproduced, and one was able to be reproduced. Not that they fake data, it's sometimes not so easy to exactly in terms of methods. So this is his idea, to make things really clear, value and open science and blah, blah. This is his thing. Then we had one module, it's called Spark. I think it was initiated actually at the Stanford University. It's a kind of a small mentoring program with little money for basic researchers. It's a kind of a, not mentoring for junior people, it's really mentoring kind of, and translates this and bring also sometimes industry in, because they want to reduce risk and they give all the stuff then back to academia. And then the last thing is, clinical research units, right, where you can translate. So there's not a single one answer to this question. And specifically for psychedelics, I would say in terms of also of guidelines, right? I mean, for 50 years, we were not allowed to do a study. I think Episode is the first one we have in Germany. I think Switzerland and Netherlands are perhaps a bit further ahead, but for 50 years. And now I would say within a year or two, I think the psychedelic field actually in the clinical research is moving relatively fast, I have to say.
You can also briefly comment. I also think there's not one solution, but to go a little further on the behavior point that has been raised, rodents are not humans, but I do think we can do much more than what we are currently doing. Like the tools have been laid out for many years, say in psychophysics and in others, and I think we can do more in having translational studies that look at comparable computational processes, like the notion of computational validity, for example, that some people are discussing in the field of computational psychiatry, where we're looking at similar cognitive processes. We can back translate, we can maybe not make some of the behaviors more, the human behaviors more animal. It's hard to translate in animals, but we could also more often perhaps go the other way around and look at behavioral studies in humans and how we can design some that might resemble more things that we see in animals. And along these lines, I think there's a lot still that we can improve here.
I mean, I think I really, to follow up on that, I was thinking what Dietmar's saying. The other, I mean, and this is the importance of various academic centers starting psychedelic science centers. David has one at UC Davis, obviously there's the Hopkins Center, is bringing preclinical neuroscientists and people studying human subject, clinical neuroscientists, together in the same physical location with shared funding will make an enormous difference. I mean, historically, at least in the United States, it's been virtually impossible for individuals to write a grant where there's two, let's call it rodent researchers and two human subjects researchers. That's slowly changing. And again, I think that's what's the excitement about psychedelic science is an opportunity to actually build those bridges where, I mean, to me, the most important thing is for people like me to talk to people like you, to, you know, to hear what's going on in the human subjects world, to pay attention to the challenges people studying human beings face, and, you know, vice versa. I think that could go a long way to building bridges, just pragmatic things like that. So it always comes down to money, funding.
Addition, I mean, we are talking about basic researchers, clinicians, scientists, or clinical research. Here is an additional factor which I found interesting. And this I thought about when Rob yesterday gave his talk. So MDMA, is there a long-lasting effect or not? LSD, is there a long-lasting effect? Most likely. But MDMA, for example, it's interesting. It's an adjunct, right? So we need also to bring in people from psychology. So psychotherapy, that's also an interesting, very important variable, I think. How, I mean, this we have to include there. And I think there's also no standard there yet, right? I mean, how to do this, when exactly and how. Mm-hmm.
What do you guys think?
I have a comment. What about employing the public? Like, I would argue that I don't have an estimate, but more than 99% of the psychedelics are being used not in clinical trials or treatment facilities, but out there in festivals, parties, raves, and there's a huge amount of people who are would be very excited to participate, not just participate, but contribute to the field. What about getting the scientists out there to the? There are some projects I heard about with Burning Man, Zendo project, but I think there's not enough of those.
No, I totally agree. But we tried it a couple of years ago. We went to a psychedelic festival in the Netherlands, and it was very difficult to get our hands on participants or to get good data out of these people because they combine a lot of things. It's a really rich field, but it's very difficult. And we were not able, we had, I think, 100 participants or something, but we were not able to get something out of the data. So another way, I also work together with Quantified Citizen. This is an app you can install for people who microdose or not, and you can engage in such studies. But then also you will have selection bias. There's somebody who wants to add something.
Yeah, I mean, I'm not an expert there, but I mean, it's an important, valid point. I have a really nice roundtable discussion with a couple of clinical colleagues, and there was one idea that we approach one of these more serious, let's call it this way, retreat centers, for example, in the Netherlands, and to see whether we can do something there. It's work in progress. It's not my expertise, but we have beside the clinicians also a psychologist, and we just started to make contacts there. Another thing I found interesting actually is I met somebody who's a very experienced psychiatrist, and he said, well, now he sees patients and they have this experience. And for them, it's really not easy to talk to them. I mean, they don't have the experience. And they also, there's sometimes, then I would say, these patients come in and they have another knowledge or more knowledge, so to say. This I found also an interesting aspect, actually. Yeah. And this guy also then, he wanted to have also such an experience to be able more to talk to the patients. Yeah.
Retreat studies, or we go to retreat studies. We do not run them. Yeah, but But the thing there is also that they actually only include people who are healthy normally, and you also want to have the broader spectrum. But also there, it's difficult to. What we like to do is follow them up, and like I showed you, you have high dropout rates also there with field work, you want to have a control group, also very difficult. We now have a control group, but it is difficult. We're trying, and we want to get the data out of the people who really do it, but difficult.
I have a question for David. I have a question for David first. When Twitter was still Twitter, so I follow your lab tweet. This is quite a while ago. I read that you have one of your new psychoplastogens in trial. Would you like to update us a little bit about this?
Yes, I can say some things. I can't say everything, I think. This is a non-hallucinogenic psychoplastogen. It went into phase 1 clinical trials. The trials have not finished, and so the data has not read out yet, but so far the drug has been very well tolerated. That's about all I can say.
Or if it's X, yeah. Thanks. So, questions? Yeah. One second. Can we have the microphone here, please?
I had the wonderful experience of attending the MAPS conference in Denver and was astonished to meet all sorts of people who told me they were clinicians and that they were doing treatment with psychedelics. And since I'm very professional conference oriented, I was like flabbergasted. What are your training to be doing clinical treatment of patients with psychedelics? Nobody had an answer, and now I see people are offering trainings in America for clinicians to do psychedelic treatment. And what are they doing? And do you have any way of assessing those people to tell them what they should be doing? I think you need to find an answer to that, because if that doesn't work and people have bad experiences, one good case in the newspapers in America and this whole field will go backwards. It's very dangerous, and that's what I'm concerned with.
Yeah, you have very valid points, but I also think that there are many underground therapists who have done this work for years and years, and we should get data from them. I have also been approached by these people, and then it's also the thing like, they don't have ethics approval to collect the data, so you cannot do anything with the data, but still we need to find a bridge there to find out what they do, to have the good practices, and then base the training on that. Yeah. It's
very frustrating.
Anyone else from the audience? Yeah. Can we? Thanks.
So my question is a little bit more about the science. I feel like one of the biggest issues facing psychedelic neuroscience is the replicability. And that, most likely has to do with a lot of the factors. The rodent strain that you use, the behavioral assays often have very small signals. But I think that's exacerbated by the fact that we're using, first of all, psychedelics, and there's multiple psychedelics that we can use. So as leaders in the field, how do you guys think that we're gonna come to sort of a consensus on these really big ideas that a lot of labs are having a really hard time replicating?
It's an incredibly important question. Issues of replicability in science have been around for 40 years. It's not a crisis, it's always been an issue. I'm gonna go back to something I said. It comes back to funders. And David probably knows about this. So I'm doing an experiment in the Bay Area with a philanthropist has given me money that I have distributed to 6 different labs in the Bay Area, 5, I think, actually, two at UC Berkeley, 2 at UCSF. These are universities in the Bay Area, 1 at Stanford. And we are all giving our mice psilocybin, same dose, same source of psilocybin, same strain of mice. And we are running a battery of very simple behavioral assays. We just got together for a 5-hour meeting for the first time to compare data to see. And I think that is the only way that kind of organized scientific enterprise, because if the funders don't give you money to do replication studies, it's going to be the Wild West. It's going to be tons of findings that that are probably going to be wrong, not because anybody is fraudulent, but because of all these variables that can't be controlled. So that's my personal feeling, is how we can start addressing it. And I think it's particularly important in this field, because these, all it's going to take is, what you said, is some bad stuff to happen, and the field's going to go back to the way it was, at least in the United States, after, in whenever it happened in 73 or something. But there may be other answers to that question.
This is also what I meant with robustness of data. That this one colleague of mine is arguing that this is the main bottleneck for the translation. So what are we personally doing about this? So starting right at the beginning of the chain, introducing electronic lab book. That was the first thing. Sounds so trivial, yeah? I had a really high-tech guy in the lab, and he said, well, you guys are still using these old books. You have all these fancy machines to write in. This was by law. It took me 2.5 years with lawyers that we are allowed to use electronic lab books. Sounds simple, but it's very transparent. Everyone can look into it. Also, later on when you publish something, that's the first thing. Then the data acquisition. You need a data manager, and it has to be open, value and open science. So we have specifically somebody hired as a project manager for value and open science. He also looks at all our methods papers. Now, the methods, that this is as transparent as possible and that people can reproduce this. And I can keep going. This is a very important issue, and then journals are looking more and more into this, and funding agencies. And this is sometimes a bit, I had this discussion actually this morning. If you put pressure, scientists, you have to do this, of course, they will do it. But I think having intrinsic motivation is even better. If you put out the data there and you have clever people looking into this data, you sometimes find interesting stuff. This is perhaps a better motivation for some of the people. Yeah.
Briefly pick up on your last point, like data sharing, and one of the things, not by far not the only thing, but one of the things that could help, because of exactly what you had said, and alongside, but that's easier said than done. It's really, really challenging. I started a lab last year, and I was thinking about this a lot, and there were lots of discussions. The Bernstein Network in Berlin, for example, they have data sharing platforms, for example. You need a lot of resources. And I realize now, well, where is the money supposed to come from? I really struggle, like getting any money to have resources for that, for example. And so unfortunately, I don't really have a good solution here. There are some ideas, they are a little bit in place. It seems there is some movement. Having open science is one example, but it's a really big problem.
You want to add a comment to that? You have your hands up?
Yeah, I want to emphasize that I think that a lot of people who have never done behavioral neuroscience, I think they look at it and say it's simple. It is really, really hard, because there are so many variables that you just can't even imagine. Even if you have like a perfect protocol, everyone follows it to a T. Maybe there was, I don't know, some construction in the hall or a loud noise, happen, when you were running an experiment. There's so many factors that go into it that make it variable. And so even if you can control for everything possible, the behavior is still going to have pretty big error bars from person to person, lab to lab, even within the same lab. We found there's differences sometimes between whether a man or a woman runs the experiment for just small little things like that. So what we've been doing instead, we still have to use behavior because that's kind of the standard for the field. We've been trying to focus more on direct measurements of circuit activation. So looking at things like immediate early gene changes, changes in neuronal structure, change in electrophysiology. What we have found is that seems to be much more replicable from person to person, lab to lab, than the behavior. And we hope that maybe we can find something that would be good enough to translate into a measurement that we could do in a human.
Like an online question that you want to...
Yeah, we have a question from our online audience. So Josh Hartman asked, do you have an opinion on Moliners et al. Publication suggesting that psychedelics promote plasticity via direct binding to TRKBs? Have you replicated their findings?
Is there in the audience, so
Why did you ask me? I mean, I wrote about that in the paper.
Who's going to take the microphone? You want to answer that one?
Yeah, I'm the wrong person to comment on that. Maybe David. I mean, I would like to hear David's comment on that.
I can't tell. Is that Euro? Is he there?
Yeah.
Yeah, that's you.
I was the one that was asking your question before.
So, I mean, I think it's a really intriguing finding. Of course, it got to be replicated. But I think, who knows? We'll see. We'll see.
I think it's time to end the panel. We guess we are at the end of the day and pretty tired and looking forward to the rest of the evening on a more social note. So thank you again, everyone. Kim, Rob, Torben, Dietmar, and David for tuning in from early morning. So I hope you had some coffee or get ready for the day. Thank you again. Thank you, the audience, for taking part.